So thank you all for connecting today. This Séminaire Général will have three main parts. In the first short part, we'll take a few moments to honor the memory of Sharon Bigley, a science journalist who recently passed. Then in the second part, I will introduce Professor Keolu Fox, who will present his research. And in the third part, I will start by asking a few questions and then open the discussion to the audience, and everyone will feel free to ask questions. So sorry about that. So the first part is about honoring the memory of Sharon Bigley. Sharon was a science journalist. I met her first at the same time I met Keolu in Berkeley. I'm guessing that some of you might have known Sharon herself. I'm guessing also that many of you read her articles. And for those who weren't able to directly read her articles, probably you heard news that was based on Sharon's reporting, on Sharon's ideas, and on Sharon's work. I don't want to make a long speech on Sharon. I will rather show a 1-minute excerpt from the interview I made of Sharon a year ago when we met in Berkeley.
Level science. Hardly any people even know basically what he did. They have a vague recollection that CRISPR babies was trending on Twitter. They have a vague recollection that they read a few stories or heard a few TV segments about it last November. But most people have no idea that that happened, let alone what it might portend. So I think people in this world might be making an error when they think that this is a topic that is top of mind for ordinary people.
And why is that? Is it because we only talk about this on a national or international level when something like Genome and the CRISPR babies happen? Or
it's because people are trying to live their lives and put food on the table and keep their jobs and keep their children from, getting shot. This is just a debate, a conversation at a level that does not intersect with the lives of the majority of people, at least in this country. Hardly any.
So I suggest that we observe a minute of silence in memory of Sharon before proceeding to the presentation. Thank you for sharing this moment with us, with me. And we'll start the presentation right now. So we are very happy, we are very privileged to welcome today Professor Keolu Fox. Keolu Fox is an assistant professor at University of California, San Diego, affiliated with the Department of Anthropology, the Global Health Program, the Halijulu Data Science Institute, the Climate Action Lab, and of course, the Indigenous Futures Lab. Before that, he was a postdoctoral fellow working with Alexis Komor. He holds a PhD in genome sciences from the University of Washington, Seattle, and one of his current research interests, and the reason why we're inviting him today, is on testing theories of natural selection by editing gene, and hence questioning the narratives that go with these theories. Professor Fox, thank you very much for being here today, and the floor is yours.
All right, aloha everyone. I'm joining you from Honolulu, Hawaii, which is where I'm from. I'm actually from a different island, one of the Leeward Islands, big island of Hawaii. And I am honored to be here, and one day I'll make it to France. And man, you guys have amazing food, amazing art, amazing culture. I guess the closest I've been to France, I've been to Europe, and I've been to French Polynesia. So
That
that's
counts.
it. Yeah. So it counts. They have bread instead of rice. Okay, I'm going to now, can you see my screen? Okay.
Yep, it works perfectly.
Okay, here. Okay, so here is the title of today's talk, which is Rewriting Human History and Empowering Indigenous Communities with Genome Editing Tools. And this is just like an aerial view of our campus, part of our campus in La Jolla, California. I'm currently not there, but it's a beautiful place. I encourage many of you to come visit, and I can talk a little bit more about that. But first, I'm from here. I'm currently here in Hawaii at the top of the Polynesian Triangle, which is massive. I think people don't really understand how big the Pacific Ocean is until they go there. Just to give you some context for many of the things we'll talk about today, a lot of my work is centered in the Pacific. I'm from the Pacific. The Pacific has shaped my genome. And we'll talk about many of those things, but we're proud to be Hawaiian, and it's another very beautiful place to go. And we have a very unique series of research programs and questions. It is a truly remarkable place. The place I'm from, it is one of the most biodiverse regions in the world. It has 11 out of 13 biomes on planet Earth. It has the most diverse soil profiles in the world. It is a remarkable place for agricultural technology development. We have every single known renewable energy resource system. It's also the extinction capital of the world. It is also the invasive species capital of the world. So it's virtually impossible to grow up on an island like this and not think about biology and natural selection in really complex ways. It's embedded in our cultural history. It's embedded in much of the language that we use to describe both the Aina, the land, and the Moana, the ocean. This is just my family, and we're cooking something called lau lau. You can see us doing it the traditional way. This is my yard. This is on the Big Island. Yep, they're delicious. And we fill them, the taro leaves, with all kinds of different food, like, you know, basically whatever you catch, right? Fish, different types of beef, pork, whatever. I know you guys are French, so you love cooking amazing food. And we have different sayings in Hawaii, and one of them I think is really important for today, and it's ika wa mamua ka wa mahope. And this just means that we're walking backwards into the future. We're identifying poor decisions that we've made in the past, not to repeat them. It explains this way that we understand the past. So when we use tools like genome sequencing to interpret our past and empower communities in certain ways, this is a really important idea. I got started as a scholar, actually, though, at the University of Maryland, and I was an undergrad student, and I studied historical archaeology. So the way I've arrived as a genome scientist is very different from a lot of my colleagues. I had the pleasure of working at the Wye House plantation and working on several excavations there, where one Frederick Douglass, who's probably the most, in my opinion, the most famous American ever. We call him the American Lion. He is very much responsible for and contributed to the abolishment of slavery in America. And we were responsible for repatriating his past and his life as a child on this plantation, where he taught himself to read and write as a child, oscillating, kind of going back and forth between the Y House, Eastern Shore, and Baltimore, Maryland. And it dawned on me how important it is in the way that we interpret material culture and we repatriate individual people's pasts that are central to really important moments in our history. And that was the first time I really got started, thinking about that and letting that kind of form a lot of the science and the scholarship that I participate in. I then went to the National Institutes of Health, and I worked at the National Human Genome Research Project first as an intern and then as a fellow. And I focused on neglected tropical disease and something called GWAS studies, which are still sort of in vogue, but we have better technology to provide higher resolution looks at the genomes. But this is my first sort of entry point thinking about computational genomics and the sort of the way these new digital tools allow us to think about the past. And I worked on this really interesting project that took place in Walaita, Ethiopia, with a postdoc, now professor at the National Institutes of Health from Addis Ababa, Ethiopia. And in Walaita, there's this really interesting gene environment interaction going on with some of the communities there who work in the agricultural settings, but they don't wear shoes. And so what happens is there are tiny microparticles of obsidian glass that's in this volcanic soil. And certain people, they create like inflammation-based podoconiosis or non-filarial elephantiasis. And what we noticed through this project is that certain people develop this and certain people don't. So it's a gene environment interaction. There's also an element of social stigmatization, because those individuals that develop this disorder, they're not intimate with people who do not, right? And so it's this runaway effect. To me, this was like my entry point for thinking about a lot of really complex ideas. And this project was really successful. Facile ended up publishing this in the New England Journal of Medicine. And that made a lot of things possible for me. One, watching a Black man dominate and do a really good job in a field where that wasn't really, obvious everywhere. And to see him publish his work in this really unique way in the most impactful journal in the world made a lot of things possible for me down the road. So I just want to thank Facile for his awesome mentorship. Then I went on to UW, or University of Washington in Seattle, is a really interesting place because it's like a tool developer academy. I mean, a lot of the tools that enabled the genomic and post-genomic revolution happened and were developed by scholars there. So Lee Hood is the kind of founder of this department, along with Bill Gates, who Microsoft, and it's part of the medical school. And their whole sole focus was this duality between what's genomic, genetics, and computer science. And so we developed a lot of tools there. While I was there, we developed exome sequencing, all of these different targeted capture sequencing, many different new genome editing tools. And I got to work on using those tools to create higher resolution blood types for hematology-based projects for blood transfusion and organ transplant. So very, very medical, very applied work. And some of the things that I was able to discover were individuals, for example, that didn't have large portions of the ABO gene itself, probably the most popular gene, the poster child, if you will, for human genetic variation. And this hadn't been seen before. Luca Cavalli-Sforza didn't see this. No one that worked in, blood group related genomics had seen things like this. And the reason is because a lot of the focus was on populations of Western European ancestry rather than kind of turning the tools and interpreting the genomes and variation of kind of historically underprivileged populations. I also worked on determining the blood types of Neanderthals and other archaic individuals from genome assemblies from, some of these new ancient genomics publicly available data sets. And that's actually work that took almost 4 years to complete, and that's coming out later this year. We're very proud of that. But using the same skill set, you can approach this and say, what's somebody's blood type based on their genome? Well, what about an ancient individual, and so on. Now I'm a professor at UCSD. We make calculated decisions. UCSD is perennially rated the #1 college for surfers by Surfer Magazine. So you can see the types of decisions that we're making. I had to move out of Seattle because it just rained too much. Cool city, but the weather is terrible. I don't care what anybody tells you. But La Jolla is lovely, and it is also an epicenter for genomic technology development. We have a really thriving private industry with companies like Illumina and others. So it's a lovely collaborative environment, and the weather is amazing. So that brings us to 3 of the research focuses that I want to talk about today. One is safeguarding ancient genomics, and this is a series of projects that we participated in and made it to the finals of the MIT Solve competition, winning the community prize, and I'll talk about that project. Then I'll talk about functionally investigating just so stories using base editing tools. And then I will kind of outro talking about building more equitable systems for managing the health data of indigenous communities, new projects that are emerging in my lab. So first, we entered this project at MIT Solve. This is a competition for like really radical, wild ideas. This is our team. It's composed of a really multidisciplinary kind of group of people, myself, one of the museum curators here at the Bishop Museum in Honolulu, our provost at UCSD, an archaeologist and good friend of mine, a political science scholar, Cameron Grimm, someone in education, Teresa, Damien, who is a bioarchaeologist, Andre, who works on ethics and artificial intelligence, and our lawyer, Ray, who's an attorney. And we focused on some really interesting ideas. A lot of them were motivated by all of the colonial behavior that's happening in anthropology, not just today, but just broadly. I should like to say and give France credit. France has been kind of at the forefront of this decolonial museum focus on planet Earth, and that speaks to the progress and the revolutionary kind of attitude that France has had historically. So very cool. But also, what is Indiana Jones doing in this scene? When I was little, this was like my favorite movie. Obviously, I studied archaeology, but I think what we're not seeing here is that we are normalizing some pretty awful behavior. And this was really motivated by this man. This is Hiram Bingham III. He was also a Hawaiian resident. He came via missionaries, and he's probably the most famous looter, grave robber, tomb raider in history. And we celebrate this, but we really shouldn't. So when we think about how museums acquire these large collections of ancestral remains or artifacts that are collected, 100 years ago or 200 years ago, we have to understand how these things end up in kind of cold steel drawers in the first place, i.e. what is the provenance in the way that an artifact is acquired, right? And when we think about the anthropology of the past, ideas like phrenology and anthropometry and all of these kinds of ideas around measurement theory, it's not that different from the anthropology of the present, where we are engaging in ancient genomics, grinding up indigenous people's bones to extract DNA without their consent, sequencing their genome, digitizing that information, and then making a lot of claims. And I think like the only way I've come to this position is that I have participated in and been complicit in these systems. So as a way to think about that and moving forward, I wanted to design tools to safeguard against this sort of bone rush and this type of behavior. This article came out a few years ago in the New York Times, and it brings up some really interesting questions about emerging technologies. And it says, is ancient DNA research revealing new truths or falling into old traps? And as an archaeologist with a background in a lot of these things, I can tell you that we're definitely falling into some of the same old traps, but we're also reimagining and providing a higher resolution of our deep past and our migratory history. So there's some amazing science going on at the same time as some serious human rights violations. And to make matters more complex, this is an article that we published in Nature, myself and John Hawks, the Fox and Hawks paper. And we determined that more ancient genomes have been sequenced in 2019 than the entirety of history. So there's this acceleration or what we're calling the bone rush of individuals that want to be involved in ancient genomics research. You can complement that with the number of ancient genomics labs and that workflow that's being constructed all over the world. In blue, we can see labs that have an ancient genomics workflow that were constructed prior to 2010. And then in red, you can see labs that were constructed post 2010. So you can see that there's this new emerging interest in processing more and more ancient genomic information. To make matters even more complex and gross, there is an emerging market of sales of ancient human remains at Sotheby's, Christie's, auction houses all over the world, Etsy, eBay, and Instagram. And so this makes it really difficult because we have a large emerging digital market where people are basically providing access to people's remains to the highest bidder, so to speak. So we wanted to develop technologies that speak to that. So one thing we've been thinking about is assessing provenance of remains in museum collections, providing detailed metadata physically, right? Like CT scans, weight before an experiment, weight after an experiment based on the gram, and then connecting that information on a ledger system or blockchain system in order to create a record of that information. So you can't just trade around indigenous people's genomes like baseball cards, and you certainly can't trade our genomes around like that either. And if we have a timestamp of when something is accessed and so on and so forth, we can have a nice record of who's accessing what, when, where, and why. I hope that makes sense. The second thing we wanted to do was really think about using artificial intelligence to interpret the deep past, but not only that, we're using something called computer vision, which is a specific type of machine learning. And this allows us to bank and compare images that we might collect and think about on something like Instagram, perhaps, where we can search for something like human skulls, and we can identify and analyze those images and train an algorithm to determine who is being involved in an illicit network of trade online. And that's exactly what my colleagues, Damian Huffer, Sean Graham, and myself are interested in doing. And that is identifying those individuals who are being involved in these networks. You can go in and even look at the individual tags. And I'm sorry if human skulls and things like this are disturbing to some of you. I apologize. But we can find networks of people that are trading things online, and you can see that there are even forms of dialogue in the way that people broker deals in the comments section. And you can imagine organizing that information and the hashtags, so to speak, in meaningful ways to deter the sale of indigenous remains. And all of that is just to say this is this larger, these are counter technologies or deterrent technologies that we're creating, but it's kind of part of this larger moment and movement within the decolonial museum studies setting. And here you see these Benin bronzes that are about to be sent home from the British Museum. Finally, all of that is to say, as a native Hawaiian scientist, we have been indigenous futurists and Hawaiian futurists. We created the first color newspapers well before France or the United States of America or any country like that. We had electricity in Iolani Palace well before the White House. Our King Kamehameha had put cannons on traditional voyaging canoes as an innovator when he unified the Hawaiian Islands, and we have been creating world-class doctors well before it was cool. So all of that is to say, we have always been using and creating the best technologies that we have available to support and empower our communities. And finally, we started a museum exhibit called Regenerations, Challenging Scientific Racism in Hawaii at the Bishop Museum. And on our debut here, you can see we had a rainbow, which is a really auspicious sign, and we're really happy about that. So secondly, that brings us to the main project that I want to talk to all of you about, which is using genome editing tools to create accountability in population genetics. And a lot of this is motivated by the sort of dearth of population genetics data that features historically marginalized communities. This is a meta-analysis of 3 different studies that I did over time, and you can see that the numbers are still dismally low. There's a really cool tool called like the Population Genetics Tracker by Mills et al. And I think the number is steadily hovering around 90% of GWAS studies exclusively feature individuals of Western European ancestry. But it gives you a pulse on that. And there's kind of 2 reasons for this. One is there's this historic kind of this history of exploitation. And so that means that people are hesitant to participate. And then there's also this kind of like convenience factor about who has access to healthcare and who's participating in these studies and who do we recruit. And so we're trying to, over time, break that down and include more diverse populations of people so we can fill in this complete spectrum of human genetic variation. One of the main issues, though, with only sequencing one population of people is it also leads to the development of something called just so stories. And if you're not familiar with this idea, it was first popularized by a children's book by Roger Kipling, who also wrote The Jungle Book, and 2 evolutionary theorists at the time, Stephen Jay Gould and Richard Lewontin, took up this idea and they said, this really aptly fits what's going on in evolutionary biology because people are using adaptionism and these narratives around natural selection to kind of enhance or popularize correlation-based science, not causation-based science, correlation-based science. And this is highly problematic because it's not rooted in fact, it's not connecting genotype to phenotype. It's a trend or a correlation that you observe. Like, how did the elephant's trunk become so long? Oh, it became really long because a crocodile tugged on it for 5 minutes, you know, something like this, right? Like real Lamarckian adaption-based story building. And it's become quite problematic throughout the evolutionary biology and human genetics community. And I want to explain a few of those examples. I hope that makes sense to everyone. Does that make sense? All good? Okay.
Yeah, I think it's good.
Again, here's this famous paper published in the Proceedings B, and this is the one where they talk about the spandrels of San Marco. It's a classic. It's one of my very favorite evolutionary biology papers. If you haven't read it, I encourage you to take a look. Okay, so let's just focus on this. So how has the environment shaped our genomes? It's a question that a lot of people are really trying to understand. It's a very interesting question that Charles Darwin, for example, was thinking about. We have not stopped thinking about this question. We have this other false narrative that mutations that are medically actionable are independent from mutations that are the result of natural selection, when in fact, they are the exact same thing. That is one just so story. But also, it's really important to think about these things. So here are 2 examples. One is genetic adaptation or genetic mutations that allow humans to thrive at high altitudes in the Himalayas or the Andes, for example. Or also Indonesian divers who've evolved bigger spleens to hunt underwater. So my question to you guys is, which of these narratives is true based on what you know? And I'll just let any person chime in.
All right, I'll go with none.
Which one?
Either.
None. None. Oh, you're skeptical, but that's you, baby. But anyone else? Florent?
I've heard this Indonesian example. I think some people may run, below the sea level at 11 or 12 minutes. So I guess this is true.
Okay, that's good. That's good. The truth is, we really haven't confirmed either of them in a functional mechanistic system. But I would say, based on the evidence of what we see, and I'll explain what I mean, that chronic hypoxia as a route to shaping the genome in high elevation adapted communities is much more consistent with the data that we have. But the acute hypoxia scenario, we know way less about. There is new data coming out in mouse models. It's going to be very interesting to see, but we really don't know. So I would say more the high elevation example. We have some really choice genes that have been identified there, but it's okay. It was a trick question, guys, you know? But on top of that, the investigators who worked on this project, they were accused of helicopter science and helicopter research. That is going into an indigenous community, collecting their genomes, extracting it, publishing that work, and not really thinking about how that impacted that community or disenfranchised them, or how that data is integrated into this larger big data sort of economy, right? And these aren't conversations that a lot of people are having with their partners, and we'll get there. But I just wanted to say, like, it's one thing to propose and propagate a inconsistent scientific narrative, but it's another thing to violate a community's trust ethically. But you can't do both at the same time. You can't. Maybe you can do one and get away with it, but not both at the same time. That's like a really poor standard of science, in my opinion. So, but let's look at some of these other examples, like the global distribution of locally adaptive traits, and have a conversation about that, because many of these are really great examples. So let's go through and kind of grade them. ABO, why do we even have ABO blood groups? Does anyone know? No? It's interesting. Also, I saw a statistic. More people know their horoscope than their blood type. How crazy is that? That's terrible. It's so disappointing.
That's terrible.
I know, it's terrible. It's awful. And that's not just America, okay? That's like, okay. But nothing has been more profound in shaping our genomes than infectious disease. Nothing. There's been nothing that's close in terms of claiming the lives of human beings over time and shaping our genomes. That's got to be pathogens. We're watching it in full effect right now with the COVID-19 pandemic. This is not the first or last pandemic. And we have these regional infections that have really had a profound impact on shaping our immune systems. But there are other examples of things, for example, high elevation adaptation, which we discussed, two really interesting genes, EPAS1 and EGLN1. These mutations are involved in something called the HIF pathway. They're really interesting and really cool, and they are signatures of natural selection for high elevation adaptation. Another one of them, EGLN1, was, and mutations there, was also described in the Andes, supporting this kind of convergent evolution idea that you could have high elevation in these two different places, and it, you know, selected for mutations in this gene in two different populations. Does that make sense? Like, that's another additional line of correlative evidence that supports that this mutation is involved in high elevation adaptation. There's another one over here, the FADS loci, which was discovered in the Greenlandic Inuit. It's a really interesting mutation that looks like it's involved in fatty acid metabolism over time from eating lots of marine fats like omega-3 and omega-6. But the verdict is still out on basically what this does and what it's involved in, and a lot of the claims. But all of this is to say, over time, we're going to discover more of these mutations as we sequence new populations of people. We just have to be very cautious about how we build narratives around them. The one that we're going to focus on for the rest of today's talk is right here. It's called CREBRF. It was discovered smack in the middle of the Pacific Ocean in Samoa initially. And it is what investigators are calling a thrifty gene, which is highly problematic. If you're familiar with the thrifty gene theory and the thrifty genotype theory, and what this mutation looks like it does is that it predisposes to obesity, but protects against type 2 diabetes. So let me say that again, because if any of you have worked in endocrinology settings, you know that you expect to see a linear correlation between protection against, obesity and type 2 diabetes. So as one goes up, I expect to see the other. I don't expect to see one go up and one go down, right? I would never see that in any metabolic disease setting. So this mutation is really intriguing, not only for people in endocrinology and people who study the Pacific, but also for big pharma, right? So let's get into it. Little bit of background on the thrifty gene for those that aren't familiar with it. It's this idea that these mutations and genes that are involved in the metabolism are really, really important for hypercaloric storage. So over time, we would select for these mutations that make you really good at storing calories, but also once you sort of move into like a modern lifestyle, predispose you to type 2 diabetes. Does that make sense? If you've not heard of this, okay. And this is from, anyway, okay. So let's think about these just so claims. And I've put like little pictures from the Kipling book over here, just thinking about these little fairy tales. The observation is that variation in this gene isn't found in individuals who are obese. The finding is that thrifty point mutations in CREBRF predispose to obesity, yet protect against type 2 diabetes. That was published in Nature Genetics in 2015. And that the selective force, this is the biggest issue. This is the narrative building at work. The selective force that maintained this thrifty allele in Polynesia is related to scarcity of calories during the Austronesian diaspora. So the media loves stuff like this. They run away with it. The Japan Times said that this is the gene that may benefit sumo giants and how a powerful obesity gene helps Samoans conquer the Pacific. Okay, you see where I'm going. This, again, is highly problematic because as a Hawaiian person, I can assure you we are the greatest voyaging, seafaring community in history. And it wasn't because we had some innate genetic mutation. It's because we're incredible scientists. It's because we utilized ecological metadata like bird migration patterns for land finding birds, different types of wind and weather systems, the green glint on the bottom of a cloud reflecting a lagoon 300 miles away. And that's all of the things we did at daytime. At nighttime, we utilized the celestial sphere, constellations, and we oriented ourselves and we discovered and pulled up islands from the sea in the most remote places in the world. That's Tahiti, that's Marquesas, that's Tuamotus, that's Hawaii, that's Aotearoa, that's Rapa Nui. But at no time did we ever have problems with provisions, right? Think about it. Caloric intake, that was never an issue. There was always pigs and chicken, fish and taro, sweet potato, all of these things that we brought around the world as incredible horticulturalists were on these boats as we were voyaging. So the whole argument just crumbles right there. You understand? But that's because there was no consensus building with Polynesian, Micronesian, and Melanesian voyaging communities in building the narrative. But making matters more complicated, we do have really, really high rates of obesity. And I don't think it really comes down to innateness. It comes down to the residue of colonialism. So think about it. If you have a community of people that is used to subsisting off the reef and you separate them from their fishing rights and their hunting rights for these really healthy foods and their, traditional way of living, and you replace it with Spam, white rice, and soy sauce, what do you think is going to happen? Right? It's going to lead to metabolic disorders because we're not eating healthy. We're not being healthy. We have high levels of stress, right? So when we look through some of the data for this paper, you can see this is where this mutation lives on this Manhattan plot. Here it comes up as a G2A, right? This specific protein change. It has an effect on body weight if you have 2 copies of it, which is the indicator for BMI and obesity. And it looks like it has this protective effect of disease when we look at the odds ratio for type 2 diabetes. When we think about the genetics and what we know about this gene, though, we know where it's located. We know there's evidence of positive selection for selection scans and FST and things like this. We know that there's a disease association, that it's associated again with higher BMI, leptin levels, but the mechanism is ill-defined, and it's very interesting because this mutation, or sorry, this gene is expressed in virtually all tissues, so it's sort of nebulous. CREB proteins are linked to mitochondrial function and metabolism, but this gene is also involved in inflammation, which is one of the most complex processes involved in common complex disease, period. That's cancer, that's type 2 diabetes, that's heart disease, etc. When we start looking at other populations besides Polynesia, not just Polynesia, but Samoa, we see that this mutation is pretty frequent throughout Polynesia. It's not just found in Samoa, and it's not found in any other population other than in the Pacific. It was recently described in the Chamorro population, who is quote unquote Micronesian. So that brings us to this other series of questions about how we geographically divide island communities. Who's Micronesian and who's Melanesian and who's Polynesian, and how did that distinction in narrative building even come into place when we know that there's frequent transit and voyaging throughout the Pacific? So the first provocation that I want everyone to think about is, how should we properly characterize the effects of these population-specific mutations? What's the best way to do this? If you're not happy with correlation-based science, how do you bring it from correlation to causation? And the way we have been doing this is using CRISPR-Cas9 base editing. So I worked with Alexis Komor. We use a modified Cas9 that is catalytically inactive, and it has tethered to it a DNA damaging catalyst. And we can, with really, really precise, accurate precision, modify individual bases and convert them. So we can utilize various base editors, and now a new version of this is called prime editing, to modify individual locations. So it's sort of like reverse engineering mutations in isogenic cell lines. Does that make sense? So we can take this mutation, this CREBRF mutation, we can introduce it into a cellular system, and now we can ask significant questions about that. Another thing that's really cool about this system, and what Alexis taught me as a postdoc, and I'm continuing to learn, is that it's extremely cheap. So rather than paying $20,000, $25,000 for Synthego or one of these companies to develop a cell line for you, we can do this for under $1,000 reproducibly. And here are those results, and these are just Sanger sequencing traces of that mutation that we've reverse engineered in an HEK293 cell line. And it was extremely efficient. And we're continuing to do this with other mutations. We're doing this with the EPAS1 high elevation adaption mutations. We're doing this with specific mutations that only exist in Neanderthals. We're doing this with mutations that look like they've intergressed from Neanderthals. So it's all about now reverse engineering the mutation in the proper physiological system to test its effect, which is pretty fun because we're playing the same ancient genomics, or sorry, reverse genomics games from the 1950s, but we're doing it with the precision of base editing. And we can ask some super cool questions. And now we can say, is it really a thrifty gene? Because you said it's a thrifty gene. We can test for insulin sensitivity, inflammation profiles, metabolomic profiles. And again, this system is easily adapted to answer a lot of different questions. It's not perfect, right? Like every cellular system has its drawbacks, but as long as you're creative enough to spend the time to figure it out and slam your head against the bench for a while, you might be able to create a cell line that's valuable for you and the scientific question you're trying to ask. And what we're seeing with working with colleagues at the University of Auckland and professional rugby players of Polynesian ancestry is that this mutation is much higher, the percentage of it in professional athletes of Polynesian ancestry. And we have our collaborators have some really cool new data that I can't talk about, but it is leading us to believe that this is more of a tall, dark, and handsome gene and less of a thrifty gene, given its sort of connection to elite athletic performance. So you see how we're building a narrative that's totally different based on empirical data, and the other one was speculative at best. Now, in terms of selection and building out the story and the why, it's probably far more likely that a mutation like this exists under selection at this percentage because of colonialism. So we're starting to see the presence of mutations and diversity or lack of diversity in gene regions like HLA as a result of European arrival in indigenous communities, a population collapse of 80% of people, causing recurrent bottlenecks and a decrease in diversity. These are things that we can detect, and making it even more sensitive for detection. And adding another line of evidence is if you compare cross sections in time and you compare HLA diversity, for example, in pre-contact ancient genomes with post-contact modern genomes in the indigenous population. It's exactly what Ripon Mali and S.K. Willerslev did here with populations in the Pacific Northwest. And you can expect a lot more of this in the interpretation of genomic variation from Cook to Cortez broadly, right? And these are scientific questions that we're really interested in my laboratory. And it's really empowering for our communities to understand the effect of that. Things get really complex though when we identify mutations like this and investigators apply for international patent claims, attempting to commercialize the exclusive use of the physiological response of alleles like this. So when this happens, you have to start asking serious questions about what and how is digital sequence information derived from indigenous communities? How is it embedded in this big data economy? And how do we find more equitable avenues for development so that our communities receive a cut of the way that it's commoditized? Also, how these population-specific mutations are embedded into surveillance systems and the violation of human rights in places like China. I think it's really important that we have serious conversations about this as much as possible, because that is not a future or a community that anyone deserves to live in. So the second provocation that I like all of you guys to think about is, should all genomic data then be open to the public? And if so, why? I read this book last year, and it's amazing. It's called The Age of Surveillance Capitalism, The Fight for a Human Future at the New Frontier of Power. And it describes how global tech companies like Google and Facebook are persuaded to give up our privacy for the sake of convenience. You can read this book. It's pretty long, but it really, really gives you the setup for like what the new economy is. What's a data-driven economy? Data is the #1 resource on planet Earth, surpassing oil in 2018. And surely it's painfully obvious when companies like 23andMe decide to go public on the stock exchange, or Ancestry.com get sold to the Blackstone Group for $4.7 billion, that this is a giant economy. And we have to have those types of conversations. We can't just look the other direction. And that brings us to the final part of this, which is building more equitable systems for managing the health of indigenous communities. And that brings us to the idea of indigenous data sovereignty and data sovereignty broadly. And that is the idea that you control your data and everything that you generate, including your genome. We published a kind of quick review on this in Nature Reviews Genetics. I encourage all of you to check it out. It's entitled The Rights, Interests, and Expectations of Indigenous Perspectives on Unrestricted Access to Genomic Data. And we've got a lot of coverage in the press about these ideas, especially around COVID data collection and diagnosis. A lot of this kind of, information is being banked because of the pandemic, and Indigenous communities are not happy about it. To make matters more complex, we have the commodification of this genomic information, which is really interesting because it gives us these unique insights into physiology that exists. So we're already seeing these private genetic mutations that are population specific be turned into the next generation of blockbuster drugs. Companies like Regeneron are well on their way to do this. We have PESCI9 inhibitors, which have been a successful new class of cholesterol drugs. They've already hit the market. There are other drugs that are in the process of hitting the market. Here's one of them. And we also had this new cystic fibrosis drug hit the market from Vertex. And in America, sadly, we have a broken healthcare system. So we have drugs hitting the market that cost $300,000 a year. This drug was actually developed in partnership with individuals who have cystic fibrosis. They use the Cystic Fibrosis Foundation, which is like a nonprofit, to recruit patients to sequence their cystic fibrosis gene and refine their drug and then sell it back to those exact same individuals. That is a violation of the common law. That, to me, is a violation of human rights. That is not the way drugs should be developed. We need to change this broken system, and I wanted to bring that to everyone's attention because it's wrong. There's no other way about it. This is not an accessible way to create drugs to deliver treatment to people. That's not why I'm in medicine. And so, based on that and finding more equitable strategies to think about the development of drugs, data as a resource, and many other things, I published this paper in the New England Journal of Medicine. And we provide strategies for thinking about data flow, current research and development partnerships between large government initiatives like the All of Us program, pharmaceutical companies, and indigenous solutions for tracking provenance and setting up equitable relationships for benefit sharing. And that could include a collective interest or community trust model where we create circular economic systems that provide percentages of royalties that go back into communities' hands to help us deliver quality education, healthcare, and cultural revitalization, or an individual interest model. In the case of 23andMe, if they do go public, why not give the people that have graciously allowed you to survey their genomes a few shares of your stock in the process? How hard is that to do? It's not hard at all. But we'll see if we can put enough pressure on these companies to think about more equitable ways to deliver benefits. All right, so the future, I'll just wrap it up. I know because I've been talking for so long, so I really apologize. More ethical relationships need to be built. I think people really think that ethics is an afterthought. It's like something you do for authorization to do a study. It's never really the origin, the impetus of the projects that people build. For me, it is, we start there and then we build out and we think about the technologies, or we build technologies that are based on ethics, but we don't append it at the end just to say we did it. You know what I mean? Like, that is the exact opposite. I think all of you know that. There's no substitute for grassroots research, co-design, and community partnership building. There's no substitute for democratizing the development of tools and technology. This is my friend Hayden. This is the first time he's doing a PCR. He's Maori, and it's just really cool to give people like fluency and transparency around how this technology works. We started the Summer Institute for Indigenous People in Genomics, and we had our first conference in 2020. This is the first International Indigenous Genomics Conference with all Indigenous speakers. Algorithm fairness, very important. Thinking about ways that we can reduce bias in the development of all types of things. There is no substitute for including vulnerable populations in the assessment of data from populations. I can assure you, if you involve communities in the process and co-design of building anything from an experiment to an algorithm to a paper to the author list, to having these communities of people in your laboratories, it's going to make your institute a better place. It's going to make for better science. It is the future. There is no denying that. And finally, remapping the human genome and thinking about new ways to provide better genomic references. What do I mean by that? The current reference that we use is derived of an individual that is majority Western European ancestry. So there's all these inborn biases because we compare all variation to that sort of key reference genome. One of my PhD advisors at UW published this paper where they created a population-specific reference for a Melanesian individual, and they literally discovered new human genes that had never been seen before. Not variation, new human genes that have functions that are immunologically relevant. So all of that is to say, there's so much more work to do in terms of really, truly thinking about the importance of diversity. It's not just a key word that's cute to use. It is extremely important in how we understand natural selection and human genetic variation. We're working on a major project around oceanic genomics and opportunities for solidarity with Hawaii, Samoa, Tahiti, Marquesas, and the Maori. And here we have this image of this He'e map of Polynesia and our interconnectedness, and thinking about not only our diaspora, but also our disease susceptibility for diseases like gout and cancer and you name it. And we've started the Indigenous Futures Lab, now the Indigenous Futures Institute. And we're working with local partners in French Polynesia and other places. And that's all, folks. And thank you very much, guys.
Fantastic. I think I'm clapping for everyone. Annabelle is clapping with me. Many thanks for this amazing presentation. Well, I'm a fan, but yeah, I think everyone agrees that it was one of the best we've had recently. So I'll start by asking my 3 questions, and then we'll open it to the audience. So I have 3 questions. Some of them you already answered in your talk, so I'll go a bit faster on those ones. The first is on the consequences of your proposal to take on this Thrifty Genes narrative for the communities that are at the center of it. The second is on how CRISPR shifts the narratives that are related to sickle cell anemia, something we've talked a lot in the news recently. And the third is on the consequences of your proposals on sovereignty, which is something we work a lot here at Sciences Po and at CEVIPOF. Okay, so the first question, you're advocating for genome editing, in this case, CRISPR-mediated base editing, but it could be something else, to be a tool for empowering indigenous communities by questioning this Thrifty Genes narrative and those kinds of narratives. How confident are you that the institutional system currently in place in the US will let that happen? How do you trust a system that has been built on appropriating human dignity to respect its own rules when it conflicts with its design? So let's take an example. We recently saw how the people behind this Robinhood app, this trading platform that didn't charge a fee for orders, have simply frozen the bleeding of predatory hedge funds that were shorting some stocks, including the one that we saw everyone in the news, GameStop. Because apparently the rules of the market do not really apply anymore when the guys who are not supposed to win do not win anymore, which means that it was never really the market it pretended to be. Now, you talked about 23andMe, now it's publicly traded. What makes you think that the communities you designate can keep control of what they revendicate as theirs and the narratives that you are trying to build around it? What makes you think that there are institutions that will uphold their rights on what makes these genomes specific? So to sum it up, when you write, and I quote your article, that genome editing can help to create a new level of accountability in the identification of global signatures of human adaptation and the narratives we build around the signatures of natural selection, end of quote, who do you think will enforce that accountability? So that's question one. Question two.
Can I do question one first? Can I do question one first?
All right, if you want. Sure, go ahead.
Yeah. Because then I'll forget, but it's a great question. I'm glad you gave me all the easy ones, yeah? So first, I'll just say that I think like it's a collective, it's a shift in focus in terms of, it's like generational, I think, personally. It's like we're starting to realize that extraction and sustainability and colonialism and all of these things are interconnected and interrelated. Like the current trajectory of science is, it's like the value proposition model is, what is this system optimized for? Well, it's optimized for making papers and making tenure and extraction and then like a light version of capitalism, right? So light version. So because we couch things in terms of the greater good, right? We're like, oh, we're doing this study for the greater good. But now we're getting into really deep questions around whose greater good are we talking about? So the more indigenous people, for example, that get a PhD or a JD or an MD that are in these spaces, the more they challenge the status quo. So it's kind of like this cumulative effect of people that will entertain these things because they have that worldview. And then we disrupt the status quo. So in the case of 23andMe, it's like they can continue to do what they're doing. But if we don't check them and people keep buying it, then what does that mean? That means that you support them. Knowing that they're going to sell your data to GSK for money and you paid them $150 for them to tell you what your mom and dad already told you about where you're from. That's crazy. That's like a crazy person's worldview. But if we create new companies that say, oh, actually, one of the companies we started, Variant Bio, it's like 4 to 8% of the proceeds go back to the community in the development of drugs. If that's every other company's model, then it forces 23andMe to do the same thing, because if they don't, then they're financially not viable. Something like that. So that's my
that's
response.
Fair. That's fair. And you had, so this was the easy one because you had talked about 23andMe before. So my second question is a bit tougher, and I have a trick up my sleeve for that one. So it's on a different narrative, gene-related narrative that has been disrupted by CRISPR, by genome editing, but CRISPR-powered one. And so this trick is a 1-minute audio excerpt that I want everyone to listen to.
I forgot to mention, I think, of course, that something ought to be done about this disease, sickle cell anemia. Here you have 1,500 children born a year with this disease and the doomed to a pretty poor life. And then we suggest that every young person have his genotype tattooed on his forehead, so that young people would recognize at first sight that they are compatible in this way. In fact, to be said in this paper, and then you see they can refrain from marrying one another, because if they marry one another, a part of their children will inherit these two genes and will grossly defect it, be grossly ill, will die young, suffer and die. But they could marry normal people.
So this excerpt is from a lecture. It's a lecture pronounced in November 1970 by Linus Pauling, who was a recipient of the Nobel Prize in Chemistry.
Two Nobels.
Sorry?
2 Nobels.
2 Nobel Prizes. Wonderful, even better. So in this lecture on molecular diseases, he argues, as you just heard, that every young person have his genotype tattooed on his forehead. It's not every young person, it's every one person that has susceptibility to sickle cell disease, so everyone who is Black. So that was half a century ago. It was still 25 years after Auschwitz, so we know what's behind the tattoos. But my question is really about the new narrative that we hear today about the bearers of sickle cell disease. So sickle cell disease, for French speakers, it's drépanocytose, c'est l'anémie falciforme. My question is, what about this hero narrative that we keep hearing about sickle cell disease people, people that have this disease, sorry. So the main argument, that's the main argument we saw in the movie Human Nature. You have this young black boy with the disease, and then you have a series of middle-aged white experts that talk about genome editing and how it's great. Then that's also what we've seen with the media push, capital-pushed media heroization of one of the participants to the CRISPR-powered clinical trials, Victoria Gray, being a hero. So, for being this part of this clinical trial. There are several narratives that can build on that, but the worst of those is if you set up a GoFundMe and manage to dry up a significant amount of your community's resources, we'll give you the opportunity to be a hero and swap up your sickle cell disease with infertility and possibly leukemia. But that's not all, because if you're a hero, then you shouldn't question why you don't have state-funded healthcare to treat that disease in adulthood, or why this monogenic disease still exists at all in the US after centuries of state-enforced policies of segregation that unwittingly probably made sure that it could still thrive in that specific community. So, second question, to sum it up, as an anthropologist, to what extent do you think those CRISPR-based therapies are a new installment of the manufacturing of expendable, combustible, now we call them essential, American heroes whose bodies are going to fuel the future welfare of those who own the machine, those who will sell those therapies, when these therapies, thanks to the trials, will be made safe and so an actual product?
Yes, a lot to unpack there. I found the Linus Pauling thing kind of dated and disturbing and very almost eugenic, but that shouldn't surprise you. I mean, look at Jim Watson, right? So where to begin? One, I mean, we've learned so much from sickle cell as a disorder about like red blood cell dynamics and mechanistically and, you know, a lot of things, and natural selection and other things. Now, one thing that's super sad is that the mechanism has been like well defined for some time, but the treatments are all often out of reach because this basically affects Black people, right? So it's not a priority for big pharma because it's not a market. And you can see how fast Pfizer, Moderna et al. Came together to provide innovation in less than a year. And I think that is a really interesting telltale sign to me of like what's our potential as a biomedical community. Like we provided a solution for something quite remarkable in a very short amount of time. And now I know Moderna is working on a sickle cell based mRNA therapy right now. And that should be hitting the market. And then that brings us to our second question, like, is it going to be not achievable? Because I feel like scientifically it's achievable to create an mRNA-based therapy for sickle cell, and that will happen. But is it going to be attainable for historically marginalized communities, given like all of these problems with healthcare and the distribution of therapy? And that's like the really depressing prospect, because you just don't know. You know what I mean? I hope that we can get there with these technologies. The hero stuff is really annoying. I mean, I find it I saw someone give a talk recently, and they had some people in Ghana wearing t-shirts that said like, I gave away my DNA, and I wanted to be like, oh, do you have t-shirts and a picture of French people wearing those t-shirts as well that you use in your presentation? So there's a lot of that, like, we're here to reduce health disparities. And I can't tell you guys how many grants I've written where I'm like, we're going to use next generation sequencing to reduce health disparities. In fact, I'll leave you guys with a story too. One time I was talking about nanopore genome sequencing at this National Congress for the American Indian in Washington, DC. And like the president of Navajo Nation was there in the back. He's at the coffee cart. And there were a whole bunch of elders. And I was talking about how this is going to like revolutionize and make genome sequencing more ubiquitous in these communities. And at the end, this older gentleman just stuck his hand up and he's like, hey, I don't really, why should I give a fuck about your gene machine when we haven't had clean water on our reservation since 1912? So like, those are the types of like disparities and issues that we're dealing with. So there's like the skill set you get in medical school that you think is going to work, that you popularize and you design technologies for, and so on and so forth. And then there's the actual problems. But if you're not interfacing with communities, then you can't co-design things that have impact also. So I hope that's.
Yeah, no, that's a very interesting explanation, and your story echoes back to the segment I played of Sharon in the beginning. Discrepancy between what we're talking about CRISPR and the actual problems in the lives of most of the population. The third question is much shorter and in a sense more theoretical. So building on the ideas that you presented, how do you link the specificities of the Polynesian genomes, all of those, to the Polynesian sovereignties? If the narratives, as you explained, around these genomes have been tools of colonial domination, then do you consider your work to be a tool or a system for reclaiming a form of sovereignty?
It's a good question. So we're creating all kinds of provenance-based tools too. Like one thing I didn't get to put in here is these ideas around record keeping and blockchain. I think like certain people that are dismissive of blockchain don't understand it. They don't understand how it is very much the future. It's such a simple tool, but it's so important for us to track the provenance and the locations of things, whether that's in a museum setting over time, or for example, I work with the World Economic Forum, and a lot of the ideas and the usage of blockchain is around counterfeit technologies. So they want to know that when you get Egyptian cotton at Target, I don't know, what's the equivalent of Target in Paris? Like,
it would be Monoprix, but yeah.
Yeah, okay. How do you know it's 100% Egyptian cotton? So what you can do is you can use a genomic-based blockchain provenance system, and you can trace the origin of that cotton all the way back to the farmer, right? And in that way, you're providing the economic system with this safeguard or deterrent technology to avoid counterfeiting, but you're also, on the other side of the coin, empowering the farmers that created it in the first place, right? So this is how we sell we sell people a product around blockchain for one reason, but it has this duality. It's like a Trojan horse, right? Because it allows us to empower and build these systems of benefit sharing behind it. So we're exploring a lot of different ways where that works with respect to genomics. So if we know a mutation has its origin in a particular community of people, then we can trace it back to them. And we can use these genomic blockchain-based systems to do this. And we're doing this with like Manuka honey in New Zealand, or, I think it's very much the future. And then the way we redesign and think about like the interoperability of healthcare systems, right? Like electronic medical health records. Who accessed what, when, where, and why? Do you have to pay to access that information? Or is it just a free-for-all like it is right now, where systems and institutions that are prepared to take advantage of open data environments are going to get richer, and the individual person whose data that actually belongs to gets nothing. So we have to really think about how to build systems around that information. So we're doing that. And one of the ways we're doing this, yeah, so anyway.
No, that's you. We don't have much time left. So thank you for answering the question. I think that the main picture here is that, and what is very surprising to me, and I suppose to my my colleagues as well, is that you're trying to weaponize capitalism in your favor, which is not something that we talk a lot in our own post-colonial research and with our own. We're more on the Marxist side when we talk on those things. We don't have many discourses post-colonial discourses that try to weaponize capitalism against different forms of institutional dominations. So that's very enriching for us to hear that. So I will now open to question. I note that Annabelle has raised her hand. If you want to ask a question, please write to everyone or to me in the chat. This way I can keep track. Annabelle, we can't hear you. We can't hear you. Sorry.
Thank you. It's the most wonderful talk. I just apologize. 1 minute it was 2:30 and the next minute it was 5:30, so I missed the beginning. Years and years ago, I was at an Institute of Science, Ethics and Innovation in Manchester. And just before I left, they had this amazing talk on Lamarckian evolution. I wanted to ask you, I'm so ignorant, but the implication of this talk was that events can cause stress in fetuses that are then heritable and so on in ways that presumably don't work on sort of more Darwinian models. And I wondered about that because I think in the talk it was all about whales or something, that but that seems really relevant to things like colonialism and the effects of colonialism, and also sort of perhaps dating differences. But I'm so abominably ignorant. Is this completely daft?
No. You're actually ahead of the curve. I think like we're getting there with the technology. So yeah, we're getting there. So you can think about these like epigenomic moments of stress and the detection of certain acetylation and methylation patterns and the transgenerational inheritance of different types of stress and how that manifests in health disparity. How does it kind of, so to speak, get baked into the genome of sperms or eggs of some, right? So I think like these are valid questions, and lots of different scientists.
I think you cut your mic, Keolu. I think you.
We lost you.
Sorry. So many different scientists are connecting this to, acetylation and methylation patterns, like what's actually empirically detectable and reproducible, and then how can we connect this to major historic events that shape or bake in these potential health disparities transgenerationally, right? So a lot of people are interested in this. I think that it's going to be a very, very cool future. It's going to be an opportunity to achieve justice using science. And you could think about a number of different historical events where this could be valuable. But designing the studies and working yourself way backwards is quite difficult. But we have one project that we're working on that is super cool. But I mean, again, so we can't. But I totally think that that's definitely an avenue for future development, and yeah, definitely.
Okay, that wasn't
not
just
a daft question. Not a daft question at all.
Okay.
Yeah. Yep.
Thank you both. Any other question on Keolu's talk or on? I don't want to monopolize the questions. Do you have?
Oh, can I ask another if there's still?
Go ahead.
Is that all right? Sorry, sorry, I'm so ignorant about these things. Can we go back to the Melanesians and the idea of what's your standard model for the population you compare things to? So I'm scientifically abominably ignorant, but I'm interested in philosophy of race and gender and all of that. So is the implication of this that really we're going to have regional specific models of genetic variation? Or is this going to go in a slightly different, perhaps a more politicized way, where instead of doing it based on region, you're going to try and do it based on, you could do it on any number of things, presumably. Were you part of the Turkish Empire? Were you part of the British Empire? I mean, where do you think that goes? Because as a political philosopher, I always want more politics than we often get in these stories.
Yeah, I think like ideally, ideally, there's so much political baggage on this, but ideally it goes to the individual level. So you have your genome, and that's how we determine what your precise therapies are in the future. That is like the end goal of personalized medicine, participatory medicine, precision medicine, all the P's, yeah, preventative. So that's like there's this gentleman, Lee Hood. He's kind of like a technological determinist and maven. He's like my academic grandpa, but that would be his take, right? The Georgia, what's this guy's name? Church take is this individual genome. I think like what is more realistic, because we're kind of in this transitional phase where we're like, we have this one genome, it's allowed us to do a lot of things, but now we're starting to see the inadequacies, all of the inborn bias that's coming out of this, because we develop every single algorithm based on this thing, and people are starting to get fed up and they're like, okay, that's racist. Like, yes, it is racist, but you have to apply new solutions. So as we move into this transitionary phase, it's like, okay, we have population specific problems where maybe having not just a genome of an individual, but a whole family, for example, because that allows us to track the transmission of rare mutations that are medically actionable. And you can think about that in terms of like the rare disease world. These like pedigree-based approaches with next generation sequencing have been extremely successful for like Miller syndrome, Kabuki syndrome, all these rare family-based congenital diseases. When we actually assess the impact of genomics on common complex diseases, cancer, heart disease, metabolic disease, it's been far less effective. And that is interesting because that's how the lion's share of people die, right? Heart disease, cancer, number one, number two. And we haven't had as much success. So I think that we want to move towards an individual thing. We need to set up population-based stuff in the interim, but it's a transitionary technology.
So because you're obviously a doctor, so you're mainly concerned with cures, but I was thinking more sort of historically and analytically in terms of what we learn about populations over history and over time and the impact that politics has.
Yeah.
And so I was thinking more, sorry, because you're quite right, we should really worry about cures rather than knowledge per se. But I was wondering whether there wouldn't be a case for going in that sort of direction too, whether or not the immediate medical payoff might be less clear.
I think they're one and the same actually. Just like with respect to the Captain Cook and Hernan Cortez thing, it's like you have a population, it has a gene pool that's like super diverse. You have the introduction of syphilis, smallpox, etc. You have a population collapse, 80%, 90%. Whoop! You have less genetic diversity. That leads to definitely the way we attenuate inflammation and other things has a direct effect on health disparity today. So yes, very much so in terms of like colonization and population displacement and things like this. And we're getting there. Like you're going to see more and more papers with folks that are, because it's really about the way you interpret the data. Because now that's my scientific question is what's the effect of colonialism on the genome? That's not something that my colleagues think about because it makes them feel uncomfortable. For me, I'm like, this is empowering.
Of course. We have time for one last question by Alistair. Please go ahead.
Thank you very much, Guillaume, and thanks, Keolu, for the presentation. I found your criticism of the just so hypotheses about thrifty genes very persuasive, and also the way you're trying to reanalyze these hypotheses seems with genome editing seems very promising and clever, but then your critique and also your earlier work in anthropology and archaeology sort of suggests other kinds of attacks on those or approaches to reimagining those hypotheses as well, which is more historical and you kind of gestured at this in the last answer you just gave as well. So with the acknowledgement that I wouldn't expect you to be able to pursue all of these different trajectories all at once in your own research, could you say a little more about whether you think there's scope, in addition to the gene editing approach you're pursuing, to kind of interdisciplinary work with anthropologists, historians, archaeologists, and so on, to flesh out what I think, again, is your very persuasive suggestion that a lot of these just so stories are based on very caricatured and simplistic accounts of the history of Polynesian peoples or those in other parts of the world, as you mentioned.
Yeah, that's a great question. So one, I would also like to say that I collaborate with a lot of people too, and they all deserve credit, and it may look like I'm doing all of these things by myself, and that's not the case at all, right? The other piece is the hope that many of you will pick up, some of the ideas and develop it in unique ways that are empowering also, and find ways to collaborate, especially in Europe. Like a lot of the exposure and connections that we have are here in the Pacific. And so it's really important, especially when we're building projects in French Polynesia. I have some really specific focus there. And I think that the more we collaborate and build on these projects, the more we kind of affect the consciousness around it. And then challenging all of these just so stories is important. And I'm not foolish. Like, I recognize that the hypotheses that we're proposing may not be perfect either. And there'll probably be some other person in 30 years that's upending what we've written. And that's the rewriting aspect of this whole thing. And that's the kind of cool self-correcting part of science and the way we do things. We're just refining it and recombining it until it's more closely mirroring the truth, if that makes sense. So, but I hope that everyone here is like either inspired or thinks I'm a bullshitter and wants to, like either way, right? Like it would be cool to develop new projects and collaborations.
I think everyone here thinks you're an amazing speaker. Paul, last question.
Yes, thank you very much. I am listening to your talk. Obviously, we understand you focus. Some of your concerns have to do with ethics and obviously so politics. Is there? Could you maybe come back on the current? Maybe there are no debates, but maybe there are current debates on like. Proposed governance on the genomes, on genetics, whether some people talk about like a global governance, a more individual governance of the genome, a more like regional, public, private. Are there any political debates, either academically speaking or even in civil society, in the US or elsewhere, concerning on how we could maybe manage those issues politically speaking in the coming years?
Great question. So this sort of oscillation around governance of data kind of alluded to this with the open and unrestricted access environment question, just to get people thinking about what the consequences of that are. Because currently it's like, it's either or, it's binary, it's 0 or 1. It's either open or it's closed. And if it's open, there's problems. If it's closed, there's problems. But there's not like a nuanced look at this that speaks to, really creating an infrastructure in between those oscillating phases, so to speak. So the open environment means that large-scale federal initiatives that use taxpayers' dollars, in the case of the US, result in open access to that data. But who uses that data? Because it's not, again, back to the greater good thing. It's like that data gets picked up by big pharma and it is basically commoditized. And you have the same people working for the federal government that enable these projects kind of operating in the same position for big pharma and capitalism. So it's a clear conflict of interest. I'm not going to say name names, but it's very obvious here in the States that it's not working for us. Then you have the individual governance model, sort of opt-in or opt-out thing. There's an idea of regenerative refusal. You could say, I don't want to participate right now until my community gets our thing together. And once our thing is together, we'll come together in a larger data trust, and we can manage this together in a more decentralized way. I think that's very much going to be the future, but right now people don't understand the value of their information. You have to think about it in terms of this is the #1 commodity on planet Earth. You can't just sit on a diamond mine and not realize it. Collectively too. I mean, we have to have more conversation. So then we have to arm our communities and educate them on futures literacy, on like where things are going. Because if you can't show them the projection, then it becomes problematic. But I hope that's sufficient. Is that okay? Is that helpful? Okay.
Yeah. We have a philosopher here in Polytechnic in engineering school is elaborating on the commodification of the intimate, so we're taking even a new turn, which is even more intimate. So the commodification of all of our particularities, it's even more takes things further. So yeah. I mean, you say the future will be bright. I hope you're right, but it makes me quite pessimistic.
Yeah, that's okay. I kind of change positions on that every day, you know. But
and we see on your fingers that the sun has risen in Hawaii.
Oh, let me show you.
Oh my God, that's amazing. Oh my God. All right. And here we are. And the night is falling on Paris.
That's good surfing. This is really good surfing.
It looks amazing surfing. Keolu, thank you so much, Professor Fox, for waking up in the night and talking to us. This was an amazing presentation and time for exchange. Thank you to everyone who connected for this seminar.